Okay, we will now continue talking about the urinary system. As I've mentioned before, the animation that you were supposed to watch covered most of chapter 24, but there were some gaps, some things that we need to fill in, and then the rest of this session we'll be talking about reabsorption of certain things a little more in detail, as well as we'll finish off talking about the rest of the urinary system, so like your bladder, ureter, and urethra. Few terms that were not mentioned by the animation or my video, my last video, were transport maximum and renal threshold. So before we get into the reabsorption of various things, we need to understand what transport maximum and renal threshold are, and they are related to each other. So transport maximum is the maximum amount of substance that can be reabsorbed or secreted in a given period of time. And that's across the tubules. So it's the maximum amount of a substance that you can move from one side to the other. Okay, so either from the tubule to the blood or the other way. Okay, if it's going from tubule to the blood, then this is called reabsorption. If it's going from blood into the tubule, that's secretion. A renal threshold refers to the maximum plasma concentration of a particular substance that can be transported in the blood without appearing in urine eventually. So we can take glucose as an example. Glucose has a transport maximum of 375 milligrams per minute. So this means that the maximum rate at which you can reabsorb glucose is 375 milligrams in a minute. Okay, so you can move 375 milligrams of glucose from your tubular fluid into the blood. And you know glucose is freely filtered, so all of the glucose actually does filter out into the tubular fluid, but you don't want to eliminate that in urine. So we reabsorb, typically we reabsorb all of the glucose. Okay, the maximum rate at which we can do this is that 375 milligrams per minute. So the renal threshold for glucose happens to be 300 milligrams per deciliter of your blood. And so that means that the maximum amount of glucose that you can have in the blood so that all of it is reabsorbed is that 300 milligrams per deciliter of your blood. That's the concentration that you can have. What that means is if you have more glucose in your blood than the 300 milligrams per deciliter, the concentration of glucose is higher. Your transport maximum is still 375 milligrams per minute. Okay, you can't increase that, but the concentration could increase, right? So if the concentration of glucose is too high, your kidneys cannot keep up with reabsorbing all of the glucose, and some of the glucose, unfortunately, will be left behind, and then it will appear in urine. Okay, so I hope that kind of makes sense. And when you look at the 300 milligrams versus the 375, these are different units. Okay, so 375 milligrams per minute, the rate, and 300 milligrams per deciliter, that's concentration. When you take into account that regular glomerular filtration rate is 125 milligrams per minute, then these numbers would make sense. So what that means is, again, at the plasma concentration of glucose of 300 milligrams per deciliter, your kidneys are reaching their transport maximum. They're transporting glucose at 375 milligrams per minute rate. If the concentration of glucose is higher, some of that glucose will simply remain in the tubular fluid, eventually down the line will appear in urine. So that's how that works. If you watch the animation, you would know that there are substances in our tubular fluid that are either going to be reabsorbed completely, and these are going to include our nutrients, electrolytes, a lot of electrolytes. Well, actually, a lot of electrolytes have regulated reabsorption. Amino acids are going to be absorbed, that's actually going to be covered in that transport of proteins. But nutrients, for sure, are going to be reabsorbed completely. The little section that talks about transport of proteins. So we said that proteins normally don't filter out from the blood, and they stay in the blood. But there is a small number of proteins that sometimes do get filtered out from the glomerular capillaries It's a very small amount, but all of these proteins, normally a healthy individual, will be transported. And we say transported because what happens to the proteins that would appear in the tubular fluid is they get taken up by cells by pinocytosis. So vesicle is going to be created, and it's going to take the protein into the tubular cell. And when that protein is enters the tubular cell, the cell will break down the proteins into amino acids. And then it's the amino acids that are going to go back into the blood. So let me show you this kind of a general image of what this would look like. And why can't I make this full screen? There we go. Okay. So what we see here is, okay, we have the tubular fluid is here, okay? So that's the inside of the tubule. And then we have our blood capillary is right here, okay? Some other terminology that we need to be aware of from this is what is a luminal membrane. Okay, we see that here. Luminal membrane is this. Okay, it's this surface of the tubular cell. It's the part of the tubular cell that is facing inside of the tubule. And then we have basolateral membrane of that cell, and that's this one right here. So that's the one that is going to be facing interstitial fluid, okay? And so when we're talking about movement of substances, I'm just going to have this up here so we can kind of see where it goes. So what happens to proteins would be they get taken by pinocytosis in the tubular cell. In the cell, they get broken down into amino acids, and then it's the amino acids that will go into the interstitial fluid and then eventually be taken back into the blood. That's what happens to proteins. So that's why we don't say that proteins are reabsorbed. We say that they're transported. So let's look at the substances with regulated reabsorption, and this is going to be a lot of our electrolytes here. And I'm not sure, you probably haven't looked at chapter 25 yet, but once you look at chapter 25, you will see that these electrolytes are discussed in chapter 25 as well. And so what I did is I kind of put the information about these electrolytes from chapter 24 and 25 together. So I need to check. I'm not sure that I uploaded the urinary system slides yet, but I will definitely, I'm making a note to myself to make sure to check so that you have the PowerPoint slides to this, but you will see information from both chapters on those slides. So upload slides or check that I have uploaded them because I'm not sure. So let's look at the first substance that's discussed in our textbook, and that's the sodium. So sodium reabsorption happens to be regulated. Our body reabsorbs between 98 to 100% of sodium normally. So your body can eliminate maximum of 2% of sodium from your body. Sodium is low in your tubular cells. So if we're looking at this image right here, we have low Na in the tubular cells, but it's going to be relatively high in interstitial fluid and in the tubule lumen. So high sodium is here. Maybe I should have used that yellow. High sodium here, and then high Na plus here, okay? So what happens to sodium normally is because we see that it's high in the tubular fluid here and low in the tubular cell, it just moves by facilitated diffusion down its concentration gradient. It's going from high to low area, okay? So this is a facilitated diffusion for sodium to go from the tubular into the tubular cell. Once the sodium enters the cell, then there are sodium-potassium pumps, and those sodium-potassium pumps are going to pump sodium into the interstitial fluid. And then eventually from the interstitial fluid, now that it's creating this high sodium concentration here, it will return back into the blood, okay? So that's how sodium reabsorption works. Sodium is our main cation of the extracellular fluid. Okay, cation meaning it's a positively charged ion. So if you don't know that, okay, it's spelled like this. It's not the red cation. I've had people that think that this is red cation, but think about cation. Okay, cation, and it's a positively charged ion. The opposite would be anion, which is a negatively charged ion. Okay, so sodium has a plus charge on it, so it's a cation. It's the main cation of the extracellular fluid. Because it makes up the most of the cations in the extracellular fluid, it is going to exert the greatest osmotic pressure, meaning that it has the strongest pull on water towards itself. And we kind of mentioned this fact when we talked about the blood pressure. Sodium, wherever sodium goes, it drags water with it. Okay, so that's how that works. The intake of sodium is generally it varies, but it is anywhere between half a gram to 25 grams per day. So regulation of sodium is pretty important, and the intake can vary. If the extracellular fluid is hypertonic, this can happen if sodium in your body increases or if your water amount decreases. Then the water would move towards the hypertonic area. Okay, so water would be pulled towards the hypertonic area. Anytime you have this concentration difference, water moves towards the higher concentration of solute. And then if the ECF or extracellular fluid is hypotonic, this can be achieved by sodium decreasing in your blood or water increasing. Okay, if you add water to your body, it's diluting things, and so that causes that area to become hypotonic. So water would be moving out of the extracellular fluid if extracellular fluid is hypotonic to the other areas. Knowing that we can eliminate maximum of 2% of sodium out of our body. If you think about what would happen if you were to drink seawater, seawater has a concentration of salt or salinity about 4-ish%. I mean, this changes depending on where we are, but that's an average percentage or average content of sodium. So if you were drinking salt water or seawater, and trying to make this a replacement for regular water, you would be constantly adding water that is too high in salinity. Your body cannot get rid of all those salts fast enough, and you would basically create your blood and your tubular fluid would be too concentrated. It would be hypertonic, and that would ultimately constantly be pulling water from your cells. So if you try to replace regular water for seawater, you would eventually die of dehydration, okay? Because your body cannot eliminate all that salt, and it would result in your regular cells losing water. So you cannot survive on drinking seawater. What else? Of the hormones that are going to affect sodium movement or sodium reabsorption are aldosterone. Aldosterone is a pretty important one, because aldosterone is going to be responsible. It directly is responsible for synthesis of sodium potassium pumps. The more sodium potassium pumps you have, the more sodium is going to be transported back into our body from the tubular fluid, or really from the cells here. Okay, so aldosterone is responsible for sodium retention. Atrial natriuretic peptide is going to be the one that limits the sodium reabsorption. So this is what is going to make the sodium reabsorption be less, because it inhibits aldosterone. So that's how it does that. Okay, so those are the important things about sodium. Let's look at water now, water reabsorption. And by the way, most of the sodium reabsorption happens in the proximal convoluted tubule, and actually most reabsorption of anything happens in the proximal convoluted tubule. Most secretion of things also happens in the proximal convoluted tubule. So most of reabsorption or secretion, most of everything happens in the proximal tubule. Okay, that's going to be kind of the trend. So if we look at water reabsorption or water movement happens by osmosis. And osmosis is movement of water. It's a passive movement of water toward high concentration of solute. Most water is going to be reabsorbed in the proximal convoluted tubule, and we call this water reabsorption obligatory. If you've watched that animation, this has already been mentioned. Okay, obligatory means that this water is pulled along with the sodium. Okay, because sodium is being moved into the tubular fluid, it takes water with it. There is about 10 to 15% of water reabsorption that happens in the nephron loop. And then in the distal convoluted tubule, as well as in collecting ducts, we're going to see facultative water reabsorption. This water reabsorption is going to depend on hormones, also on how hydrated you are. So this can change. But in the proximal convoluted tubule and the nephron loop, it's more or less the same rate of reabsorption of water, no matter how hydrated you are. Okay, it's going to be the same rate in the proximal tubule and the loop. It's the distal convoluted tubule and the collecting duct is where we can see modifications to the reabsorption. The main hormone that has to do with water reabsorption is ADH, or antidiuretic hormone. ADH is going to bind to our principal cells, and this is going to cause aquaporins to increase in numbers. Aquaporins are water channels. Okay, the more water channels you have, the water you can reabsorb from the tubular fluid back into the blood. And then aldosterone also affects water reabsorption because it is going to affect your sodium reabsorption. And if aldosterone increases sodium reabsorption, it also increases water reabsorption because sodium pulls water with it. Okay, so those are the two hormones that have to do with water reabsorption. The next substance we're going to look at is potassium. Potassium is the main cation of the intracellular fluid. So it's the opposite of sodium. And this is kind of an interesting or important thing to remember, that sodium and potassium, they move in opposite directions. Okay, so if sodium is being reabsorbed, potassium is being secreted, and vice versa. About 98% of potassium in your body is found inside of cells in the intracellular fluid, and only about 1% would be in the extracellular fluid. The sodium potassium pumps are the main pumps that are going to maintain this gradient here. Potassium imbalances are the most lethal of our imbalances, electrolyte imbalances, because potassium is very important in neuromuscular function, so it can lead to cardiac arrest or respiratory arrest. If you remember when we talked about the electrical events that happen at the heart, and when we looked at what happens at the nodal cells and how how the pace is determined, how the pacemaker cells work, there was an importance of calcium there. I mean potassium there. Calcium was there too, but potassium was important. So it just happens to be that potassium is the one that happens to be most lethal if it's not balanced. Potassium also is different from the other electrolytes, or from like sodium, for example, because potassium can be both reabsorbed and secreted. So that's just something different that we see about potassium. Where sodium, we didn't really secrete sodium, we just didn't reabsorb sodium. So because we said that sodium reabsorb 98 to 100% of sodium, right? That means that if you are only reabsorbing 98% of sodium, that means that the 2% are not reabsorbed, but it doesn't mean that they're getting secreted. It's just they just remain in the tubular fluid. But potassium can move in both directions. It can be secreted actively into the tubular fluid, or it can be reabsorbed back. It is regulated by aldosterone. Remember, aldosterone is responsible for formation of sodium and potassium pumps. And so it regulates potassium as well. Okay, that's all we're gonna say about the potassium. Let's look at the calcium and phosphate, and we talk about these together because most of the calcium in our body is stored in the form of calcium phosphate in our bone, and some of that is also in our teeth, but majority is going to be in our bones. So 99% of calcium in your body is going to be in your bones and teeth as calcium phosphate. The physiologically active calcium is in its ionic state, and what that looks like is that's calcium ion, which is Ca2 plus. Okay, out of the this is what you find, that's that physiologically active version. The 50% of your calcium that's in the blood, so not the calcium, the most calcium that's in your bones, but there's some calcium in your blood normally. 50% of the calcium that is actually in your blood is in this state, in the Ca2 plus. Some other calcium versions of calcium that can be found in your blood are when it's bound to the phosphate. So calcium phosphate, about 15% is in that kind of solid state in your blood. Probably on the way to the bone. And then about 35% calcium in your blood could be bound to albumin. But most of your calcium in the blood is the physiologically or the biologically active version in just the free ionic form. The physiologically or the biologically active calcium gets filtered out out of glomerulus right here. Okay, it becomes part of that tubular fluid, and when we compare that to phosphate, most of phosphate gets filtered out by glomerulus. So you have about 90 to 95% of phosphate that becomes part of the tubular fluid. We can regulate the blood levels of calcium by parathyroid hormone. So parathyroid hormone, we mentioned when we talked about the hormones that parathyroid hormone does regulate calcium. And so one of the targets for the parathyroid hormone are the tubular cells, where the parathyroid hormone is released in response to lowered blood calcium. And what it does is it will inhibit phosphate reabsorption. Okay. If you inhibit phosphate reabsorption, it means that phosphate stays here, PO4 3 minus, stays in the tubular fluid. It doesn't get reabsorbed back into the blood. And what that means is that calcium that's in a Ca2 plus state can remain in that Ca2 plus state. It's not binding to phosphate if we're not moving phosphate to it. Okay. So by inhibiting reabsorption of phosphate, we're allowing more calcium to stay in that Ca2 plus, independent state. So that's calcium and phosphate. The next substances are bicarbonate ions and hydrogen ions. These are going to be very closely related to pH regulation, okay? Because bicarbonate is a weak base, hydrogen is an acid, and so their levels are going to affect your pH in your blood. Bicarbonate is filtered freely. So during filtration right here, you're going to see HCO3 minus moving out of the blood. But most of the H plus stays in the blood because most of the hydrogen ions are bound to some other molecule. So they are not being filtered out. And so what happens is your blood has a potential to become acidic or too acidic, so you're reabsorbing a lot of this bicarbonate kind of right away back into the blood so that it can balance out the hydrogen. So the way these two works, if you have an acid, you offset it by a base so that it gets neutralized, and vice versa. If you have a base, you neutralize that with an acid. Okay. So we reabsorb bicarbonate because it's the one that is moving freely into the tubular fluid. A person can be in acidic or alkaline state. This really depends on your diet. So fruits and vegetables put you in a more alkaline state, and proteins, consumption of proteins puts you more in an acidic state. And that's because of what molecules our digestive system breaks this down. So byproduct of protein digestion are acidic molecules. So most of people in the United States are in a more acidic state, and that's because our diet is based largely on protein. Okay, so that is the bicarbonated hydrogen. The next thing that we need to talk about is elimination of various things. So we eliminate, and this is the opposite of reabsorption. Okay, so we talked about the reabsorption of or dysregulated reabsorption. Now we're going to look at things that are eliminated, and that means that the direction of movement is from blood into the tubular fluid. Okay, now we're going to see what is being secreted. So we eliminate or secrete nitrogenous wastes, and there are three main types of nitrogenous wastes. We have the uric acid, which is a result of nucleic acid breakdown in the liver. We can talk about creatinine. This is from creatine breakdown in the muscle. And urea is also a nitrogenous waste, and urea is formed from protein breakdown in the liver. With the urea, though, about 50% of urea is reabsorbed in the medulla of your kidneys, and it helps create the concentration gradient between the tubular fluid and the interstitial space. I know this wasn't really mentioned and usually it is not mentioned when we talk about the nephron loop, but urea is one of the substances that helps make the interstitial space more concentrated. So it helps with water movement, especially in the nephron loop. And so those are the nitrogenous wastes. Some other things that we eliminate here are certain drugs like medicines, but also illicit substances. Some other metabolic wastes like urobilin. This is from bilirubin breakdown, which is part of the hemoglobin breakdown. There are some hormone metabolites as well as some hormones that are released from the blood into the tubular fluid and eventually appear in the urine. Probably the most known hormone would be the pregnancy hormone HCG, human chorionic gonadotropin. This is how women can detect whether they are pregnant. It's that pionistic hormone. But other hormones get eliminated here as well. Going back to the reproductive system, the luteinizing hormone would be eliminated, especially if you have a spike of it, and that's what is used to detect ovulation. Okay, so some hormones are eliminated through our urinary system. So if you are following with the textbook, then it goes into the nephron loop and vasa recta, but this was part of the animation, so I want you to use that animation to really go over, how these things move and such. But some things that were not mentioned there, it talked about the glomerular filtration rate, so you should know what that is from that animation, and basically how much of filtrate you make here, okay, per minute. Okay, how much fluid is pushed out from the glomerulus per minute. And I mentioned at the beginning that it's 125 milliliters per minute is the regular average glomerular filtration rate. We measure glomerular filtration rate with a substance known as inulin, and inulin is going to be administered to a person so that the blood plasma concentration is 1 milligram per milliliter, and inulin is not secreted or reabsorbed, so it is filtered, but it just remains in the tubular fluid, and then you can measure out how much of that inulin is exiting the person's body, and then you can figure out what the glomerular filtration rate is based on that. The renal plasma clearance was not mentioned by the animation as well, but the renal plasma clearance is how much plasma you can completely clear of a substance in a given time. And this also, so it's the amount of plasma that you can clear of a substance per given time. And this tells us whether the substance is reabsorbed or secreted. So you can use that number to say if it's getting, which way the substance is moving. So that's the renal plasma clearance. And yeah. Okay, the next part of chapter 24 is dealing with urine characteristics, transport, storage, and elimination. So this is kind of heading towards the end of chapter 24. For the urine characteristics, I want you to know what is a typical urine composition. So urine is mostly water, about 5% of solids, but this changes based on how hydrated you are. The more hydrated you are, the more urine you would be producing, and the less concentrated it would be. Okay. Some of the normal solutes that are found in urine include some ions such as magnesium, calcium, sodium chloride, things like that, ammonia as well, or ammonium ion. Nitrogenous waste like urea, uric acid, the ones that we said that are eliminated, and some hormones would be there as well. The normal pH of urine is between 4.5 and 8.0, so urine can be acidic or basic based on what's found in it, and really kind of depends on your diet too. Kind of a side note here, but some women believe that they can predict what the gender of the baby would be by, peeing into a baking soda. And really, all that is detecting is whether your urine is acidic or basic. So if your urine is acidic, the baking soda will bubble, I believe. Yes, baking soda is big. Anyway, so I think it will bubble when it's acidic. And if your urine is basic and you pee into a baking soda, then it's not going to do anything. I guess the idea behind this is that they say that your tastes are different when you're expecting boy versus when you're expecting a girl. So they are saying, okay, I don't have scientific data on this, but they say that women that are expecting boys are craving more protein-rich foods, and therefore their urine would be more acidic. And women that are pregnant with girls are not craving as many proteins. Again, this is just kind of an old wives' tale. I don't know. But somebody monetized this, and you can actually buy a test in like CVS or Walgreens or Walmart or wherever. It costs about $35, and it's based on the same kind of a background where it really just turns the different color based on the pH of your urine. So just FYI for that. So that's that. The average volume of urine is between 1 to 2 liters, and it to be more the more water you drink, and the less water you drink. But on average, it should be between 1 to 2 liters. If it's less, if you're dehydrated, your body will try to retain water to maintain blood pressure at an appropriate, and healthy level. And so what you would observe is that urine will have a darker color. The more dehydrated you are, the darker urine is, because it has less water per all the solids that need to be eliminated from your body. So if your urine is getting darker and darker, you need to drink more water. Specific gravity refers to just density of urine. So since urine is mostly water plus some solutes, specific gravity would be, and I guess, I don't know if we need this picture anymore, but I'll have some other picture. But specific gravity is basically, oops, I'm still looking at the picture. It's the density. So since urine is water plus some solutes, the specific gravity has to be higher for urine, but it's just a slightly higher. It's never going to be lower because it's always water plus some other stuff. I've mentioned the color of the urine already, and then the term urinoid we use to describe the normal smell of fresh urine. So urine does have a smell. If urine is let to stand, it will eventually produce ammonia odor because ammonia is produced there. So the people that have ever seen a cloth diaper that has been sitting for a day or two in the wet bag, you open that wet bag and there is going to be smell of ammonia there because that urine breaks down into this. At which point the pH of urine would change as well. So that's but urinoid is the term that we use for the normal smell of fresh urine. The smell of urine changes based on your diet as well. Probably the most famous food that affects your urine smell is asparagus. It does have a typical smell there. Also, certain drugs, certain medicines can affect the smell of the urine as well. So that's that. The next section, we are going to be talking about the urinary tract, so ureters, urinary bladder, and urethra. If you look at the ureter, let me find the image here. Oops. So you can kind of see what that looks like. And let me share the screen with you. Here's what we're looking at. This is a cross section of the ureter, and each kidney has one ureter. A ureter has a muscular layer there. If you notice, there's a muscularis layer right here. 2 layers of muscularis there. And what I want you to understand is that the way that urine leaves your kidneys and moves through the ureter is part of it is gravity dependent. So if you're in an upright position, then the urine would flow, from top to bottom in the direction, as the gravity is pulling it. But that's not the main force. The main force is actually this muscularis layer that is pushing the urine constantly down the ureters towards the bladder. So and ureters are retroperitoneal as well. So these would be running along your posterior abdominal wall. A lot of times if people are experiencing kidney infection or kidney stone or something, it might present initially as back pain, okay, because these are retroperitoneal organs, so it feels like you feel it in your back. So peristaltic contractions of the muscularis layer move the urine from kidneys to the bladder consistently and constantly. And the lumen of the urinary bladder it really is like this. It's kind of you see these folds right here that allows the ureter to expand as the urine is entering it. Okay, it's kind of esophagus was a little similar to that as well. So it allows for expansion as urine is filling it. Okay, this would be an empty ureter that we're looking at here. So that's the ureter. So 1 per kidney, retroperitoneal, and it contracts in peristaltic ways to push urine down to the urinary bladder. Urinary bladder is a muscular organ. This muscular organ is going to be a temporary storage for urine. So the ureters are constantly moving this urine down to the bladder, and bladder is filling up, right? If you look at the layers of the bladder here, there is a muscularis layer there as well. I do want you to know that this muscle is called the detrusor muscle. Okay, so the detrusor muscle, remember that one. The bladder is retroperitoneal as well. So notice where the peritoneum is like laying on top of it. Okay, it's only covering that fundus part, the top part of your urinary bladder, but it's not going around this way. Okay, so all these parts are retroperitoneal, and there's a region. So the way that the ureters connect to the bladder is if you're following where the ureter is, it's going like this. Remember that this is posterior abdominal wall, and then it turns kind of towards, it turns interiorly and connects to the urinary bladder like right here. So it doesn't connect at the top here. Okay, this is not where it's actually touching or connecting to the or emptying into the urinary bladder. It's here. So it goes behind the urinary bladder, down below the top part of the urinary bladder, and connects kind of in the mid region here. Okay, so the openings of the two ureters and the opening of the urethra, these are what mark this area that we call trigone. It's just the region of the urinary bladder that has a name. Okay, and let me see. Do we have? We don't. If we had a different view of this, you would actually be able to see better how the ureter connects. Okay, so bladder is a little more anterior. The ureters will run along the posterior abdominal wall and then turn anteriorly and connect to the bladder from the posterior side. Okay, I guess if you look at this bottom part right here, this is an empty bladder versus filled up bladder. So this is a side view. This is a sagittal view. Okay, if this is the ureter, okay, it connects to the bladder like this. Okay, because this is a side view, right? So this is the posterior abdominal wall here, the ureter, and then it turns and connects like this, turns anteriorly. Okay, so that's how that connection works. Okay, so after there's enough urine in the urinary bladder, or whenever you decide to urinate, because you can actually consciously regulate this, the urine would exit the bladder through the urethra, and urethra, this would be a female urethra, I believe. Well, it looks like a female urethra. Okay, we are looking at, if you look at this is going to be the male urethra on the next slide, like right here. Okay, but urethra is a tube that is going to lead urine from the bladder outside of the body. There is an internal and external urethral sphincter. The external is within the urogenital diaphragm for both males and females, and the internal is right here where the urethra begins. Okay, it's at the junction of the urethra and the urinary bladder. The internal sphincter is involuntarily controlled. So this one relaxes when the micturition reflex kicks in, or if you are consciously attempting to urinate. The external sphincter is voluntarily controlled. Okay, so this is again, like we saw with the digestive system, the internal was involuntary, the external anal sphincter was voluntary. And when I said the kids are learning to use their external sphincter when they're getting potty trained, same thing with urination. Okay, it's gaining the control of the external urethral sphincter because internal is involuntarily controlled no matter what. So in females, the urethra is much shorter. Okay, much shorter than what we see in males. And this is one of the reasons why females are more likely to get urinary tract infections. If a bacteria gets to the urethra, it has a short distance to travel and get into the bladder ultimately and cause infection. In males, the bacteria would have to travel a longer distance and often would be eliminated before the bacteria can actually cause anything. Okay. So in females, the urethra is pretty straightforward, short tube that only serves for elimination of urine. That's it. That's its only function. However, in males, the urethra has two functions. It has a function that is elimination of urine, but it also has a reproductive function. So it serves in the reproductive system in a passage of semen through here and out of the body. Okay, so it carries both semen and urine. And in males, the urethra is longer, and it actually has different parts. So first part of urethra in the males is going to go through the prostate gland, and that's called the prostatic urethra. That's right here. The part of the urethra that goes through the urogenital diaphragm, that's called a membranous urethra. Okay, we don't call it membranous urethra in females. And then the rest of the urethra that's going to go through the length of the penis, that's called spongy urethra, or sometimes you might see it referred to as penile urethra as well. And unfortunately, I don't know why this picture is split, but these are with the second part of it. Okay, so in your textbook, this is all one long picture. It won't fit on my screen here. So it's divided into two. We'll be touching upon the urethra again when we talk about reproductive system, which is the next part or next unit, really. So that's that. Now let's start. I've mentioned the micturition reflex. So the process of urination depends on two reflexes, and the two reflexes are the micturition reflex and storage reflex. So micturition reflex is when it's basically kicked in whenever your bladder is about 200 to 300 milliliters filled with urine. So as the bladder stretches, it's receiving urine, right? Bladder is receiving urine, and as it's receiving urine and it fills up, it stretches. And this stretching of the bladder, when it's about 200 to 300 milliliters, initiates this micturition reflex, which causes you to feel the need to urinate, to go empty bladder. Kind of an interesting thing, though, is that if you choose to ignore this micturition reflex, it kind of goes away for a little bit until it gets filled with additional amount of urine, and then it kicks in again. Now, you cannot do this forever because once the volume of urine in your bladder reaches about 600 or so milliliters, micturition or urination will happen. Your bladder cannot store urine forever. Those sphincters, the external sphincter will simply give out. You won't be able to hold it anymore, and urine will come out. Okay, and it's not good to retain urine and ignore your need to urinate because it wears out that external sphincter. Okay, so when you feel the need to urinate, it's go and don't overwork that sphincter. Another reflex that's important in urination is the storage reflex, and storage reflex is the one that allows you to retain urine, so that as the urine is entering your bladder, it's not simply just leaking out. Okay, so that's the storage reflex, and you can also consciously control urination, so you don't have to wait for the micturition reflex to kick in. You can urinate by using Valsalva, contracting those muscles. You can kind of squeeze the bladder, relax that external sphincter, and urinate. I try to convince my children that this really is true every time we go somewhere. Well, not that we're going anywhere right now, but anytime we go on a trip, what do we tell to our children? Go use the bathroom, okay, before we get into the car, because I'm not stopping 5 minutes after we left. And they'll be like, I don't have to go. And what do we say? I don't care. You still need to go. And so that's because we can voluntarily cause ourselves to urinate. Okay. And so that's really the end of chapter 24. So it was migration. And the next part that we'll be talking about, I guess that's the Monday's thing, is the fluid and electrolytes. So there we'll be talking about like the distribution of fluid, different compartments, as well as a little bit more about electrolytes and acid-base balance and imbalance. So that's what's coming up after this. So this concludes chapter 24, and let me know if you have any questions.
Hello.
Hi.